discussed about sideroblastic anemia. So in the sideroblastic, the first basics are important. First, we should know the structure of hemoglobin. So hemoglobin has a heme portion and globin portion. Globins we have two alpha chains and two beta chains. Whereas coming to the heme portion is made up of a porphyrin portion and the ferrous iron portion. Now let us see how this porphyrin has been made. Usually this porphyrin ring keeps the iron at its center, and each chain has at its center there is an iron molecule embedded with the help of supporting ring structure, that is porphyrin. So first we should understand how to make this porphyrin ring. So the story starts within the red blood cell. Within the red blood cell we have cytoplasm and mitochondria. The pre-red blood cell, before erythrocyte, a nucleus containing red blood cell is called as erythroid cell. In the erythroid cell, first we have mitochondria. In the mitochondria we have glycine and succinyl-CoA. This glycine will be transported somehow, that we'll see in the next. So this glycine succinyl will be converted to aminoleuninolenic acid in the presence of an enzyme called ALA synthase. Furthermore, it will go for prophylinogen, methylbilase, uroporphyrinogen, coporphyrinogen. So these are the series of steps to make a porphyrin, and then it will make a heme. Whenever there is an issue with any enzyme, there will be a defective porphyrin problem, porphyrin synthesis issue, leading to improper utilization of iron. Due to this improper utilization, why this improper utilization happened? Due to some mutation in the enzyme. The defective enzyme activity lead to iron accumulation inside the mitochondria. Due to iron accumulation, there will be a reduced erythropoiesis production, leading to iron overload in the circulation. Plus, it also triggers microcytic anemia, and this iron will also cause issues with pancreas, liver, spleen, and heart tissues. Okay, this is the pathophysiology of sideroblastic anemia. When due to this, the characteristic thing about sideroblast is this one. This is a sideroblast. It is a erythroid red blood cell with a large nucleus surrounded by iron deposits surrounding the nucleus. This is called as a sideroblast. Then we discussed about the what else we discussed? The classification of sideroblastic anemia. Sideroblastic anemia broadly classified into congenital and acquired based on the cause etiology. So congenital we have X-linked, autosomal, Pearson syndrome and thymine. So these four are divided based on the enzyme issue in this pathway. For example, X-linked sideroblastic anemia is due to issue with this starting enzyme leading to iron overload and hepatosplenomegaly. Then autosomal recessive, here there is an issue with the glycine transport into the mitochondria leading to the second cause, that is autosomal recessive sideroblastic anemia. Then third one is Pearson syndrome, where there is an issue with the mitochondrial DNA. Obviously when mitochondria haven't worked properly, you will have issues, and the issues especially seen in just born babies, that is called severe anemia in infancy. Then thymine responsive anemia, where this sideroblastic anemia can be responsive. It will respond to the vitamin B12 supplementation. So this type of sideroblastic anemia can be treatable, but others are very tough to treatable. Then coming to the acquired sideroblastic anemia, we have primary and secondary. Secondary are due to some dietary issues such as diet, nutrition, toxins and poison. And primary acquired, to understand primary acquired, you should understand the stages of cancer. The first, increased cell growth and increased cell count is called as hyperplasia. After hyperplasia, they will go to metaplasia, where the cell will change its morphology. Then it will change, there will be a disorganized cells, disorganized growth and abnormal cell count, that is called dysplasia. After dysplasia, you will get a neoplasm, uncontrolled cell growth leading to a cancer. It can be a benign or malignant. Benign means something that is treatable, localized, can be removable. Malignant, game over, we can't treat it. Then coming to the dysplasia, various types of cells, uncontrolled growth and the various proportions. This is what's seen in the primary acquired sideroblastic anemia. Based on that, we have multiple things. Single lineage dysplasia, either in lymphoid lineage or myeloid lineage. Then we have PMDS RSA. What is this? Primary myelodysplastic syndrome with ring cell sideroblastic anemia. Is it primary? Yeah. Yes, primary. Primary myelodysplastic syndrome with ringed sideroblastic anemia. So that's it. Then coming to the lab diagnosis of sideroblastic anemia. So we have CBC, PBS, peripheral blood smear, then bone marrow aspiration and the genetic testing. In complete blood picture, we will go with the HB, decreased HP levels, decreased MCV levels, MCH levels, MCHC levels, and increased RDW levels. That means the red cell distribution with levels. And in PBS, we will see two important things. One, sideroblast cells. Second one, basophilic striplings can be seen, where the granules of the basophils will be stripped off. Looks like a striplings. Hence, this is called basophilic striplings, which is also seen in the bone marrow aspiration. Okay. And last and final, genetic testing will be done for various genes that are coding the enzymes in this pathway. Thank you so much. Yeah.